Dear viewers, this video is section 2, part 1 in the 5-section series on germs, friends or foes in the Medicine for Anyone in the YouTube channel. Bacteria. As free-living organisms, bacteria have all the cellular machinery they need to grow and divide independently. They are between 1 and 10 microns in length and most contain a single chromosome. When stretched out, this coiled circular molecule of DNA reaches to about 1 millimeter and carries up to 8,000 genes which code for all the proteins bacteria need to survive independently of other life forms. Bacteria reproduce by binary fission, which involves making a copy of their chromosomal DNA and then simply splitting in two. Vibrio cholerae, as one of the fastest growing bacteria, can accomplish this feat once every 13 minutes, and even the slowest growers, like Mycobacterium leprae, can double their numbers every 14 days. Given ideal conditions, as a single bacterium could produce a colony weighing more than the Earth in just 3 days. But fortunately, conditions would be far from ideal long before that. Bacteria are masters of survival, and when adverse conditions come along, they are generally ready. Adaptability is the key to their success. Yet, in theory, reproducing by binary fission yields offspring that are all identical to the parent, a process that apparently leaves no room for variability. Errors in replication of DNA so common and frequent in bacteria, basis of evolution. But although their DNA copying machinery is accurate, mistakes occur which are corrected by a cellular proofreading system. Even so, occasional errors slip through. If the errors are serious enough as to make life impossible, they die. If the errors are not serious enough but has no relevance to daily activities, then the error is retained but left unnoticed. If the errors are not serious enough but is found to be useful to daily activities, then the error is retained and incorporated into the active cellular machinery. These heritable changes to the genetic code, known as mutations, may cause changes to their offspring. This is the basis of evolution by natural selection. So, dear listeners, by the way, this educational video is made possible by Prescribon, the makers of e-prescription software for digital prescriptions, available freely for doctors on the website www.prescribon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. In humans and other animals, evolutionary change is a slow process because of our long generation times. But for bacteria, which reproduce very fast and have a less effective DNA proofreading system, rapid change by mutation is their lifeline. A single bacterial gene mutates at a rate of one change per 10 power 4 to 10 power 9 cell divisions. So, in a rapidly dividing colony, many thousands of mutants are thrown up. A few of these mutations will confer a survival advantage, and these progeny will then quickly outcompete their rivals and come to dominate the population. Gene swapping by plasmids. Bacteria have several other tricks to help them adapt rapidly to a changing environment, mostly involving gene swapping. Many bacteria contain plasmids, circular DNA molecules that live inside the bacterial cell but are separate from the chromosome and divide independently. They supply their host bacteria with extra survival information and can pass directly from one bacterium to another during conjugation. This involves the outgrowth of a filament called sex pilus, which acts like a temporary bridge between the donor male and the neighboring recipient female bacterium, giving plasmids free access, allowing survival genes to spread rapidly through bacterial communities. Several genes that code for antibiotic resistance, allowing bacteria to survive in the face of antibiotic treatment, are carried on plasmids, and they have succeeded in spreading worldwide. In contrast to phages, plasmids exist independently of the host chromosome and are generally non lethal. When the phenotypic features of the plasmid are not required for bacterial survival, the plasmid will exist only in a small minority of the total microbial population. Bacteriophages. Another way that genes can jump between bacteria is by using viruses called bacteriophages or phages for short. All viruses are cellular parasites, and phages commandeer the bacteria's protein making machinery to generate thousands of their own offspring, most of which carry a copy of DNA identical to the parent phage. But around one phage in a million mistakenly picks up an extra piece of DNA either from the bacterial chromosome or from a resident plasmid and carries it to the next bacterium it infects. If this extra piece of DNA codes for a protein that improves survival, then natural selection will ensure that the offspring of the recipient bacterium will prosper at the expense of others. Sometimes, phages set up long term symbiotic relationships with their host bacteria, with the phage being safely housed inside the bacterium, and the bacterium in turn being protected from infection by other more destructive phages. Remarkably, the toxin that can fatally damage the heart and nerves during a diphtheria infection is coded for by phages in the bacterium Corynebacterium diphtheriae. Note: diphtheria. This is an acute, highly contagious bacterial disease causing inflammation of the mucous membrane, formation of a false membrane in the throat, which hinders breathing and swallowing, and potentially fatal heart and nerve damage by a bacterial toxin secreted by the causative organism Corynebacterium diphtheriae. Another phage resident in the bacteria Vibrio cholerae causes the catastrophic diarrhea of cholera. Without their phages, Corynebacterium diphtheriae and Vibrio cholerae are harmless. Note: cholera. Cholera is an extremely virulent disease that can cause severe acute watery diarrhea. It takes between 12 hours and 5 days for a person to show symptoms after ingesting contaminated food or water. Cholera affects both children and adults and can kill within hours if untreated. Some microbes found a home in other living things. At some stage in the distant past, groups of resourceful microbes found a niche in or on the bodies of other living things and evolved to parasitize host species. From that time on, the struggle for survival has shaped the evolution of both parties. On occasion, a comfortable symbiotic relationship developed, like, for example, the microbial communities that form self sustaining ecosystems in the guts of their hosts. For ruminants, such as cows, the advantages of this partnership are obvious. The microbes are bathed in nutrients and protected from the outside world while they digest the cellulose in plant cell walls, which cattle are unable to do for themselves. Note: ruminants. The ruminants comprise the cattle, sheep, antelopes, deer, and giraffes. These are mammals that chew the cud regurgitated from its rumen. Most ruminants have a four-chambered stomach and two-toed feet. Ruminants are hoofed, herbivorous grazing or browsing mammals. They are able to acquire nutrients from plant-based food by fermenting it in a specialized stomach prior to digestion, principally through microbial action. And in humans, however, the function of gut microbes is not so clear. We each house up to 10 power 14 microbes, weighing in total around 1 kilogram and outnumbering our own body cells by 10 to 1. So far, more than 400 different species have been identified which probably protect us from attack by more virulent microbes, aid our digestion, manufacture vitamin B12 for us, and stimulate our immunity. They are harmless as long as we are healthy, but if they manage to invade our tissues, perhaps through a surgical wound, they can cause nasty infections. Of the million or so microbes in existence, only 1,415 are known to cause disease in humans. But despite their significance to us, these pathogenic microbes are not primarily concerned with making us ill. The sometimes devastating symptoms they produce are really just a side effect of their life cycle being enacted inside our bodies. However, they certainly use each step of the infection process to their own advantage, and natural selection ensures the microbes that induce disease patterns that are best designed to assist their reproduction and spread survive at the expense of their more sluggish siblings, that is, fellow microbes. So, over time, disease patterns have been sharply honed by evolution to ensure the survival of the causative microbes. A highly virulent lifestyle, killing the victim outright, is not advantageous to microbes as they will then be without a home and probably die along with their host. Yet, less virulent microbes risk being rapidly conquered by the host immune system, and this also curtails their spread. Over centuries of coexistence of microbes and their human host, evolution has fine tuned the balance between these two extremes to optimize survival of both species, but the rapid adaptability of microbes means that they are generally one step ahead in the ongoing struggle. So, dear viewers, this is the end of video section 2 part 1 in the 5 section series on germs, friends or foes in the medicine for anyone in the YouTube channel. Thank you for watching.